and welcome to another episode of the Afro Talk podcast. This is a podcast series hosted by the European Atherosclerosis Society Young Fellows. And my name is Sanna. I'm from the RWTH Aachen in Germany, and I will be your host for today's episode. And Professor van der Sluis is a professor of molecular genetics and translational biology at the University Medical Center in Groningen in the Netherlands. His research at the UMCG delves into the molecular mechanisms that control lipid metabolism and the endo-lysosomal transport of cholesterol and receptors, both in health and disease, with important implications of conditions like fat liver disease and atherosclerosis. And he's today joining us for the Atro Talk. So, good morning, Professor van der Sluis, and welcome. It's a real pleasure to have you with us today. And we are looking forward to hearing more about your career, your research, and your views on the future of atherosclerosis and related fields. And on behalf of the Young Fellows, we would like to thank you for accepting our invitation.
Well, thank you. Thank you for inviting me. It's a real honor. It's a great pleasure to be here. Good morning also.
Yeah, so in this series of interviews, we want to give the listeners the chance to get to know you a bit better. And personally, as well as on research and academic career. And so we would like to ask you to introduce yourself to the listeners. So who is Professor van der Sluis?
Yeah, who am I? Well, my name is Bart van der Sluis. I'm a father of two children. Emil, a boy, he's 12 years old, and a girl, Lucia, she's 10 years old. My name of my wife is Dineke Verbeek. She's also professor at the University Medical Center of Groningen. I met my wife in the lab in Utrecht, where we were both working as a PhD student. And we are both trained as a geneticist. And after my PhD, I moved to Washington, D.C. to do a postdoctoral researcher at NIH. And after 1.5 year, my wife also moved to NIH for a 6-month stay. And she were working on an extra project at NIH, so during her PhD. And I must say she was very successful in those 6 months, so she did pretty well. And I think almost 2 years after I moved to Washington, D.C., I moved back to Utrecht, also as a postdoctoral researcher. And in 2008, I started my own group in Groningen. Yeah, that's very brief, maybe. In addition to my work, I love to stay in the mountains, both in the summer and in the winter. In the summer, I like to hike with my family, and in the winter, I like to ski on piste with my family, but also off piste, so free riding, skiing, and backcountry skiing. And I do that with friends and a Dutch guide who lives in France. In the Netherlands, I like to cycle on my racing bike, and I also do some running. But I must say, I have to do that more frequently lately. So I have to work on this. So maybe this afternoon I have some time for running here along the river.
Yeah, we have some beautiful weather today, so
Yeah,
I think
yeah,
it will
yeah,
be something nice to do.
yeah.
Yes. So you already shared some things about yourself, but we're also curious if there's something surprising that your students might not know about you.
Yeah, that was a tough question. To be honest, I don't know. I think I'm pretty open, so I have not really that much surprises. And if I have, maybe I want to keep it like that. So
That's okay.
I can still surprise them as well. But I don't think I have that many secret things or that they don't know about.
Can you then maybe describe a typical day in your life, if there's such a thing, and how you balance also the research responsibilities and leadership responsibilities that you have?
These are tough questions, I must say. Well, in general, I get up pretty early and then I make breakfast for myself, but also for my children, prepare their lunch boxes for school. But at work, I have a lot of tasks, not only being a scientist, but also I'm a co-head of the laboratory of pediatrics, of the Department of Pediatrics. I'm also running a mouse transgenic core facility. I have to teach. So I have to keep everything in right balance, and that's not always easy. And so what I try to keep the balance between management responsibilities and science is to block 2 afternoons for myself to focus on research. But I must say that's not always easy because my agenda automatically fills it quickly fast. So I have a great help with my secretary, and yeah, she's helping to be more strict on that. So I keep those afternoons for myself. But I must say, it's not easy. So I have to work on that a little bit better.
But I think that's a good tip also for our listeners to
Yeah.
really block your time.
Yeah.
Yes. And among all the tasks that you have, do you have one that you really look forward to or that you really enjoy?
Yeah, what I still really like to do is to walking in the lab and looking at data, talking with the students and with the postdocs and discussing with them about the results, but also about science in general. And if it's possible, I also still like to do some experiments by myself, although I don't have that much time anymore. But I think a few years ago, I still did some experiments in the lab. And I think also still have the skills to do it. Probably the students will say no, but I think I still can do it. But I cannot pipette those 384 well plates. I'm not good at that anymore. So
stay away from these.
Yeah.
And do you, like you already said that you have a lot of different tasks and responsibilities. Do you maybe have some advice, like how to manage this time-wise, and how to have regulate these multiple responsibilities that you have?
Yeah, that's pretty tough. And I remember when I had an interview about my promotion to professor, they had the same question about how do you manage all these responsibilities, and I couldn't really answer that. So I said, yeah, just do it. Yeah, I managed to get it done. The only what I think is important, as I said before, keep enough time for research. So block time in your agenda to do that. Learn to say no once in a while, and be strict on that. And also, you don't have to solve the problem by yourself or immediately. You can also try that the person that comes to you solves the problem. So then you don't have to do it by yourself. But I must say, it's tough. But yeah, we try to manage these things and everything. But I think it's going okay.
Yes.
Yeah.
And yes, thank you very much for sharing these tips, also to delegate a bit task and not keep
Yeah.
everything with yourself. And now we get to know you a bit more. We would also like to ask you to tell a bit more about your academic journey.
Yeah. Yeah, so I studied biology in Utrecht, and then I did my PhD in the lab of Siska Wijmega. She's a well-known geneticist here in the Netherlands, but also worldwide. She's pretty well-known. I was her first PhD student that graduated in her lab. So after that, I worked at NEH, did a postdoc as a postdoctoral fellow. My research topic was identifying novel players in leukemia. In 2004, I came back to Utrecht, did a second postdoc, work on a mouse model that I generated at NEH. So I had the opportunity to generate my own models at NEH and took them with me to Utrecht. And in 2008, I started my own group. I was asked by Martin Hofker, who moved from his lab from Maastricht to Groningen. And I must say, Martin Hofker is also a well-known scientist in lipid metabolism and atherosclerosis. Sadly, he passed away 10 years ago. And so that's a little bit my career. And now working on lipid metabolism and atherosclerosis. But maybe we will discuss that also a little bit later.
Yes, exactly. Yes. Yeah, because throughout your career, you have explored a wide range of topics. And so you really went from the copper how do I have to pronounce it?
Toxicosis.
Toxicosis gene in the Wellington terriers. And then you also went to leucomonogenesis and hematopoiesis. So you really like have a wide range. So what motivated you to shift eventually to atherosclerosis and lipids, lipoproteins, and then cardiovascular diseases from this epidemiological perspective? And in which way did your previous research experience contribute to the understanding of approaches in studying cardiovascular diseases?
Yeah, that's an interesting story, I must say, how to move from being geneticist, studying copper toxicosis in the dog breed, and then going from leukemia to lipid metabolism. And I must say, this was unexpected. I did not really force that direction. But this was the phenotype, what we found in the mouse model I was working with. So the focus of my project when I moved to Groningen was mainly on the regulation of NF-κB signaling, so a big player in inflammation. And my interest was to better understand how NF-κB is regulated, and especially during fatty liver disease. And then when we were characterizing the mouse model, we found a lipid phenotype. So these mice had high levels of plasma LDL cholesterol levels. And that was also the research interest of Martin Hofker. So there was an immediately good match. So with these unexpected findings and with the connection of Martin Hofker, I shifted the focus of my research from inflammation more to lipid metabolism and cardiovascular disease. So yeah, that was well fitted in the lab of Martin Hofker, and but also I think it's a really interesting field of research, especially the big problem in our society about the number of people that still die because of cardiovascular disease. So I think it's also really relevant. So yeah.
Yes. Yeah. Yes. So you also worked at the National Human Genome Center Institute in the US that you already shared with us before as a postdoctoral researcher. So we would like to know, like, how was the research approach different there compared with where you, like in Groningen, where you're now? And what was your most valuable experience that you brought home?
Yeah, it's a good question. Well, in general, I think the research approach is not that very different than here in the Netherlands or in Groningen. But I must say, I had a lot of freedom at NIH. And so you have a lot of opportunities to do your research, to do all kinds of experiments. There are many, many different facilities, good facilities, with great people who can help you. And yeah, I think the most valuable experience, I think, is to take that opportunity, use those facilities, use the knowledge of other other people, and yeah, don't be shy and go for it. But really practical, I must say, I think the experience that I got working with mice, because now I do a lot, most of my research is with mice. So yeah, I got my first experience there, and yeah, I still took that with me in Groningen. So that was perfect.
Very valuable.
Yeah.
Yeah. And when did your determination to work as a scientist develop? Was this already from a young age that you were interested in science, or this is during your study?
Okay, yeah, no, it wasn't during my study. I think my third year, when I studied biology, there were some lectures that I really liked. It was mainly about cell biology, and yeah, I really liked that. So that was, I think, yeah, I want to understand that a little bit better, how the cell is working, how organelles is working, and yeah, that was, I think, the yeah.
Wanted to know more.
Yeah.
Yeah.
That was the trigger. Yeah.
Also, you shared already a bit before you some names, but we
Yeah.
would still like to be still curious, like throughout your academic journey, like which mentors had a significant impact on your mindset and approach in research, and which key lessons or valuables did you took from these mentors?
Yeah, I must say Martin Hofker. Of course, Siska Wijm also had a big influence on my scientific career, and she helped me a lot. But I think for the next step as a postdoc, and also and I wanted to become a more independent scientist. Yeah, Martin Hofstra is a really good mentor, so he helped me a lot to give me the right direction, and also how he set up his lab, because he said, well, we have to work as a team. And be open with your collaborators. Even if you work on the same topic, it's better to be open and share your data and then work together on it instead of trying to be competitors. And yeah, work as a team. Together you achieve much more than by yourself. And even in good but also bad times, it's better to work as a team because you can, in bad times, you can help someone else or someone can help you and the other way around. So yeah, I think that was, the most important lesson that he taught us.
Yeah. Is this also something you share now with the next
Yeah,
generation of
I
researchers?
try to do that. Yeah. Hopefully, I manage that as well.
Yes.
Yeah.
I think that's a very good thing to do. Could you maybe tell us a bit more about a new project that you're really excited about? So we also saw that from the Dutch Research Council, you got recently awarded funding for exploring processes involved in liver growth after birth and following liver injury. So we thought this was very interesting. Would you be willing to share some insight into this project and some findings and how they also might translate into clinical practice?
Yes. Yeah, I was a little bit surprised when I saw this question because you did your homework pretty well. So there's no secret for me, so no surprises. Yeah, so one of the proteins we are working on is that this protein is not only involved in cholesterol metabolism, but it seems to also be involved in the regulation of specific signaling pathways. And these pathways likely are involved in postnatal liver growth. And at this moment, we want to identify those signaling pathways because it seems that these pathways are also involved in liver regeneration and liver cancer. So if we hopefully find this signaling pathway, we can help better understand how liver regeneration is regulated, but also how liver cancer develops, and opportunities to identify new targets to treat liver cancer. So, and I must say, at this moment, a PhD student is also working on this project, and next week he's doing an experiment, partial hepectomy, in our mouse model, and hopefully we'll identify whether this protein is indeed also involved in liver regeneration. And I'm really excited about this experiment. And I hope everything goes well because it's not an easy experiment. So, and he's not doing that in our lab, but he's staying for 2 months in Barcelona to do this experiment. So hopefully it goes well. And that electricity is still keep going next week.
Yes.
Not
I think you chose
shut
a
down
good
as
week.
last week. Yeah.
Yes. Yeah, so we're recording this podcast just after we had like that for the listeners and like that we just had like the power outage in Spain. So now it's good indeed that you planned it now and not like 2 weeks ago.
Yeah.
Yes. So you already like shared now that you have some interesting experiments coming up, but we were also curious like the technologies that you're using. So are there any particular tools or methods you're especially excited about right now?
Yeah, so a few years ago, 1 year ago, I introduced a new mouse model in my team, lysotech mice. And these mice allows us to isolate intact lysosomes from different organs. And in our lab, we mainly focus on the liver. And a postdoc, very talented postdoc, Chrissy Vizel, is working on that right now. She also got an ES grant for this. She will receive today, I think, this afternoon. And we already get very interesting data out of these mice. So I think it's a really nice tool. Yeah.
sounds exciting. Yeah.
Yeah.
So during the opening today, you will get the grants.
Yeah.
Yes. So you're expert in lipid metabolism, cholesterol, and cardiometabolic diseases. How do disruptions in the intercellular cholesterol trafficking contribute to the pathogenesis of cardiometabolic diseases? And what therapeutic avenues are being explored to mitigate these effects? Can you share something about this?
Yes. Well, first is that the liver is the key organ in maintaining cholesterol homeostasis, systemic cholesterol homeostasis. And the endo-lysosomal system plays an essential role in this because it's essential in the hepatic uptake of LDL cholesterol and the transport of LDL-derived cholesterol from the endosomes, lysosomal system to other compartments in the cells, such as the ER, where cholesterol is sensed and where transcription regulation is regulated. So any defects in those pathways have a big influence on the uptake or the transport and eventually cholesterol synthesis and eventually, systemic cholesterol homeostasis. And it's well known and also based on our research, if something goes wrong, you see that plasma cholesterol levels goes up and increase the chance of atherosclerosis. So so a key pathway in regulating cholesterol homeostasis. Further, it's therapeutic targets. These pathways, I don't know, but it's interesting to tell. And that is a recent publication. I think it was in Cell Metabolism, where they show that the proteins that we're working on in our lab, you can stabilize that with a drug. And in this paper, they show that if you stabilize this protein, fat accumulation in the liver is going down after diet-induced fatty liver disease. So there might be some chances that you can target this pathway to alleviate cholesterol accumulation in the liver and maybe also eventually in the circulation. But we have to see.
Mm-hmm.
Yeah.
Yes, still enough to explore.
Yeah.
So, like, has your approach to science changed over the years? So you, for example, also shared about the mouse that the mouse model that you now created, which of course gave you new avenues to study.
I must say when I moved to Groningen, because also I'm head of the Transgenic Mouse Core facility, so I have immediate access to mouse models. I can generate them also for myself. And based on that, my approach has changed a lot.
Mm-hmm.
So I mainly do in vivo work, and also I often start with a mouse model before I start with cellular models, because I had the experience that you can do a lot of research in cells, and then you find nice things, and then you go in vivo, and you don't see that anymore.
Mm-hmm.
And so based on that, I often start with the mouse. If I don't see phenotype, then I immediately stop. If I do see phenotype, then I continue, and then also we use cellular models to understand the mechanism. So I go a little bit, I think, in a different way.
Yeah.
But I think it's a little bit luxury to start from mice and then to cells.
Yeah.
I must also say that the last few years also started to do more screening, set up screening systems to identify novel lipid genes. I do this with a direct colleague of me, Jan Albert Kuiverhoven, but also collaborator Noam Zelser from Amsterdam. And I must say also, I do this kind of screening also in mice. So I do not do that in cells, but I really like to do that in mice in vivo to preserve the complexity of organs and also lipid metabolism. And I think have a bigger chance to really identify good new players in lipid metabolism.
Yes. Yes, so indeed, you mentioned that you are running this Transgenic Mouse Clinic at the University Medical Center of Groningen. So what mouse model is currently in demand? And with the technological development in recent years, are there any trends in animal models in atherosclerosis research?
What I see that most people are now more interested in knock-in mice, so mice carrying a certain mutation that links to human diseases. I think in general, knockouts and conditional knockouts are still popular. But now with CRISPR, it is much easier to generate those models. So that's, I think, this is a trend. And I must say, also in my own team, but also in our department, somatic gene editing is very popular. I also see now in more publications and also in related to atherosclerosis. So somatic gene editing means that you can knock out your gene of interest after the mice are born. So with CRISPR, you can knock it out. So you don't have to generate a lot of lines to study your gene of interest in a specific organ. And the most popular organ is, of course, the liver, and easy to access with viruses or other delivery systems. And I see this is becoming more and more popular, not only in my own team, but also in the atherosclerosis field. Because it's relatively efficient, quick, and cheap, and you don't have to keep many different lines. So I think within 3 months' time, we can already generate a large group of animals where we knocked out the gene of interest in the liver.
Yeah.
So in the old days, you needed almost a year, 1.5 years before you have these mice.
Yeah. That's, it speeds up the process a lot.
It speeds a lot. Yeah. But also, we must say, you take a bigger risk and then you say, okay, let's knock out this gene and this gene and this gene. So, and
maybe push it too far.
Yeah.
Yeah, so like this is already a big step that we can generate these mouse models way quicker. So also when we look ahead, where do you see most promising opportunities in inflammation and cardiovascular research?
Yeah, I have to be careful now because, and also honest. To be honest, I don't think that inflammation is the target to focus on to treat cardiovascular disease, but that's my own opinion. Also, when you look at the CONTEST trial, there is an effect, but it was not really striking. And I think we still should focus on lipids, ApoB containing cholesterol-rich particles, because we know that's the main risk factor to develop atherosclerosis. So I think, we should still focus on that. There are many targets to lower lipids in circulation, but I think there's still room for more because some of them are still very expensive. And I think, if we find other targets that lowers lipids, it will be only beneficial. But also we should also focus on prevention. Start with lipid-lowering drugs sooner, eat healthier. And I think that's also a task of the government and the food industry. Because they keep us eating bad food and exercise more. But that's easier said than done, of course. So
yes,
but I think this is the way, I must say. But hopefully I not offend now people that works on inflammation, because it's still an interesting field of research, of course, and it has a role in atherosclerosis. I'm not saying that, but I think it's difficult to target it.
Yeah,
I think, but
yeah, but I think it's a very valid point to also highlight the lipids.
Yeah.
Yes. And good that you're already planning your own run in the afternoon.
Yeah.
And on a more general note, are there any upcoming projects or research areas that you're excited about, and especially those that bridge basic molecular research with clinical applications in hepatology and cardiometabolic health?
Yes, I already mentioned that a little bit about a project that I'm really excited about, and that's about the lysosomes and the dynamic regulation of lysosomes under different metabolic conditions. And as I said, 1 year ago, I introduced this model, and a postdoc in my group, Christy Vizel, starts to work on this project, and she has already very exciting data. And for one of her projects, she's received these ES competitive grants. And in this project, she focused on sex difference in hepatic cholesterol handling and in relation to lysosomal function. And what I did not expect, although there's a really strong difference between male and female hepatic lysosomes. And it was really striking. So she has the data already, and she put them next to each other, and it's stunning. And did not expect to see that much difference between males and females lysosomes. So I think, yeah, this is pretty exciting. I think now it will be very challenging to further investigate these differences and really show that this can explain the difference between cholesterol handling in male and females. But yeah, we have to think clever how to study this in a good way. Whether it really helps metabolic diseases, cardiometabolic diseases, I'm not sure, but maybe it helps us to understand the difference between males and females in terms of metabolism. And I think that's already a big step, and maybe that can help us better understand why there are differences in response to medication. So
Yeah.
we will see. But I think, yeah, as a basic scientist, I think it's already very challenging and very excited about this project.
Yes.
Yeah.
Yes. Yeah. Gender, like sex,
Yeah.
is very important
Yeah.
to include
I
in
think this is the
research.
first session tomorrow, right?
Yes.
Yeah.
Yes.
Good.
Exactly. Yeah. We have the sex differences
Yeah.
session tomorrow morning. Yes. So you became the head of the Laboratory of Pediatrics in 2022. Can you tell a bit more about your projects that you have here?
Yeah. As the head of the Laboratory of Pediatrics, the main task is to keep our section financial healthy. So not really keeping science or running different projects, scientific projects, but more financially healthy, keeping the infrastructure in our lab up and running, and also, of course, also keeping the people working in that they are happy in our department and motivated. So that's the main task I have within the laboratory for pediatrics. And this moment, we're also looking for ways to attract new talent scientists who hopefully becoming the new leaders in our department. So we're looking for ways to get money and to how to do this because the it's always about money and it's not easy to get enough money to attract those people and keep them because we need then also the money from the university and it's not always easy.
No. So very different tasks next to
Very
you.
different
Yes.
tasks. Yeah.
Yes. Very
Yeah.
diverse. Yes. Okay, now I would really like to thank you. We're coming to the end of this interview, but we always want to close off with a special section, which is called the Graveyard of Ideas. So most of the time we only hear success stories of researchers, which makes some of us question ourselves. And we want to all researchers to know that failure is part of the process. And so we decided to ask everyone in the series of the interviews to contribute to this idea of the graveyard of the ideas. And this could be a story about a grant that you were passionate about, but did not work out, or any kind of failure that's faced during your academic journey. So what is your contribution to our graveyard?
Oh, it can be many. I must say, not scientifically or experimentally wise, I think everything went pretty well. But I think, it's about grants that I did not get, uh, and I think that most of the time that the grants, the most important ones that I did not get because the reviewers did not believe my hypothesis or that my uh, but at the end I was still right. So all the things that I wrote down was correct. So that's a little bit disappointing that although you know that you were right and that you did not get the grant. Um, but yeah, it's okay, and then that's how it goes. But I must say the one grant that I did not get and that can have a big effect on your career. And luckily, it came out well. But that's the Dutch grant in Veni. This is a prestigious grant for young scientists, and that helps them to the first step in their scientific career. And I think that would make my life a little bit easier when you got this grant. Although it is what it is. Eventually, um, it worked out pretty well. And yeah, also I said great help from my mentor in Groningen and Martin Hofker. So yeah, eventually everything went well. So if you still believe in your own data and you go for it and keep going, then things will go fine.
That's good to hear.
Yeah.
So thank you. And within this podcast, we also want to inform our listeners about the possible open positions in different academia institutions around the world. So do you usually announce your open positions, or would you encourage researchers looking for a PhD or postdoc position to directly write the labs they're interested in? So how does this work in your institution?
Yeah, so in the case of an open position in our institute, then I post it on LinkedIn, and then people see that I have a position available. And the best way is to find on our website of our institute if there's a position available, yes or no. That's how it goes, because then it goes via system, and check the website of our institute. That's the best way to do. And or look at my LinkedIn.
Follow you at LinkedIn.
Follow my LinkedIn, and then so we have now 2 positions available, 1 PhD position, and we will start with interviews very soon. So the application procedure is closed now. So we go for the interview now soon. And I filled in a postdoc position last week in my team. So that is also closed.
Yes. So the moment you're set.
Yeah. And hopefully we get more grants soon, and then we can hire more people again.
Yes.
Yeah.
So this was an incredible interview. Thank you, Professor van der Sluis, for sharing your time and insights with us.
Thank you for having me. I really liked a lot and having this interview and this podcast.
So also a big thank you for the listeners. This podcast is for you, and we also need your feedback to tailor it to your interest. So please don't forget to leave your impression or suggestions for topics that you would like to hear. And stay tuned for the next episode. And again, thank you very much to Professor van der Sluis.
Thank you.